Hi, let's discuss about the cell cycle. There are two main purposes to the cell cycle. Organisms grow bigger by adding more cells, and if cells die off or they get damaged, we could use the cell cycle to grow new cells. The cell cycle creates identical daughter cells, meaning they're the exact same as what the parent cell was. There are four main phases to the cell cycle: G1, S, G2, and M phase. The first phase, G1, is the vast majority of the time the cell spends its life, and it's just growing in size. S phase is when DNA is copied, DNA is replicated. So when they make two new cells that have identical DNA, well, we got to copy that DNA. It happens in S phase. How is that DNA copied? Well, DNA replication. The left side of a chromosome is one DNA molecule. That right side of that chromosome is an identical copy of the DNA molecule. So you notice it's two copies of the same DNA code. So why is DNA copied? Well, if our original cell, the parent cell, had 46 chromosomes, the two daughter cells, the two cells that it creates, also have to have 46 chromosomes. So obviously the math does not add up. And of course, what has to happen is we got to double the amount of chromosomes in the parent before it splits in half. G2 phase is just basically preparing for mitosis, preparing to divide the cell. And then mitosis is splitting the cell in half to create two identical cells. There are four main phases to mitosis: prophase, metaphase, anaphase, and telophase. The actual splitting of the cells is called cytokinesis. The big picture: G1, we grow the cell and it gets bigger in size. In S phase, we copy the DNA. So you notice it went from 46 DNA molecules before S phase to then 92 DNA molecules. And in G2, we prepare for cell division, prepare for mitosis, and in mitosis, M phase, is when the cell splits in half. And you notice it makes two identical cells that have both 46 chromosomes for humans. I hope this helps. Thanks for listening.